Okay, well, thank you very much, everyone. Tonight we have a lecture which I'm really looking forward to because I know nothing about sponges. The title of the lecture is Reproduction in Sponges, Genes, Structures and Ecological Patterns. And I'm thrilled that Dr. Ana Riesgo, who's a life sciences researcher at the Natural History Museum in London, is going to give us this lecture. So she's based in London. I've just heard that she's been here for 2.5 years, and before that she actually worked in Barcelona and in Harvard. Her research spans from transcriptomics to electron microscopy approaches to various biological and ecological questions, including speciation, adaptation, and genetic connectivity. In other words, extraordinarily broad, very multidisciplinary. So I'm really looking forward to hearing your talk, Anna.
Thank you very much for the introduction. I'm really pleased and honored to be here. It is truly an honor for me to be here. I'm from Spain, so I never thought that Darwin was going to listen to my talk. But here he is. He's going to listen about sponges. And I'm really overwhelmed by the audience here, not from the portraits, but from you guys. So I hope you enjoyed this talk as much as I enjoy sponges. So we'll be talking about sponges, but first I want to tell you why sponges are important. I'm pretty sure you all know about coral reefs and corals are really important, but sponges are way more important. They are everywhere. They are super abundant, and they are very frequent everywhere, not only in deep sea, where you can see these beautiful animals, but also in temperate habitats, in tropical mangroves. They are incredibly abundant in Antarctica, in coral reefs as well. They are actually taking over the coral reefs. And they are super abundant as well in freshwater systems, both in rivers and in lakes. So they are everywhere. But what do? They provide three-dimensional structure so other animals can use them as house. So you can see one of these barrel sponges in the Indo-Pacific absolutely covered by crinoids. So they really use it as a structure. But it's the same thing in glass sponge reefs in the deep sea Pacific Ocean. They are absolutely full of annelids, of shrimp and crabs and fish, and everything lives in sponges, which is incredibly annoying for a geneticist like me. But it's very good for the ecosystem. And they are incredibly important, and this is something we learned relatively recently, because they drive major biogeochemical cycles in the ecosystems. So until 2013, we didn't know that actually sponges are the major players in the carbon cycle. So the cells that they have and they shed, they release into the water, are the actual food that feeds the rest of the organisms in coral reefs, which are really oligotrophic. And people were just wondering what happens. Why are they so diverse? It's because there are sponges. They are also major players in the nitrogen cycles because they are incredibly full of bacteria. And these bacteria are very active in the nitrogen fixation processes. So they are as well very important for the nitrogen. But most importantly, they are super, super important for the silica cycle. Before, it was only thought that diatoms were super important for the silica cycle because they actually recycle silica really, really quickly, and they deposit the silica in these fruit tubes. But sponges put their silica into the skeletons, and it doesn't dissolve. So it goes directly, again, to the bottom of the ocean. So they act as a silicon trap. So ecologically, you see, they're really important. But they are threatened everywhere. They are suffering from major diseases in the Mediterranean, in Australia, in the Caribbean, everywhere. And these diseases are related to the global warming because there are many different pathogens that start growing in the sponges when the water gets warm. They're losing habitat because of coastal development, because of mining, because of fisheries, trawling, etc. So they are really affected by all these human impacts. So if we want to protect and conserve them, we need to study them more. We need to know about their distribution, about their evolutionary history. We need to know about their population dynamics, about their reproduction. And of course, we need to understand what are the threats for sponges and how can sponges survive to these threats, if they can. We don't know. And in this sense, it is really important that we start studying adaptation. This is something that has never been done in sponges. We don't know how they can cope with global warming or any other major threats. But today, I'm just going to talk to you about reproduction in sponges. And I'm going to tell you about how sponges maintain their populations. They can do it by sexual reproduction, by regeneration. They are they have fantastic regeneration capabilities. When I was doing my PhD, I had to cut small pieces of the sponges. And after 1 week, you go there and you don't even see the scar. They regrow super fast. And they, of course, they do a sexual reproduction. So if you go to a textbook, a classical textbook of sponges, you would see that sponges mainly reproduce by asexual reproduction. Well, that's not true. They do it majorly by sexual reproduction. So I'll be telling you a little bit about the evolution of sex just in sponges, about morphology of gametes, where these gametes come from, what are the molecular toolkits that sponges use to produce these gametes, and what are the ecological implications of having different types of yolk or different larval types, because there are major differences whether the sponges choose one route or the other one. I don't know if you can see this very well, but this is just an introduction to the different classes of sponges. We have 4 different classes of sponges. The first one is calcareous sponges, calcarea. They make their spicules using calcium carbonate. But the rest of the sponges, they make the spicules using silicate. These are the homoscleromorpha. They were discovered from a long time, but only in 2006 or something like that, they were erected as the 4th class of sponges. These are the beautiful hexactinelids, or glass sponges. They as well have silicate or silica skeletons. And finally, the most abundant and diverse of the sponges, the demosponges. So just getting into the sex part, sponges can be gonochoristic. That means they have separate sexes. So one sponge would be making the sperm, and the other one would be making the oocytes. And usually, these sponges tend to be oviparous, or we thought that they tend to be oviparous. So that means they release their gametes into the water, and that's where, there in the seawater column, it's where the fertilization takes place. But there's another option. Sponges can also be hermaphrodites, so that means the same individual can make the sperm and the oocytes. And but the fertilization takes place outside the body, so there is not like auto-fertilization. And these sponges tend to be viviparous, so they brood their own embryos, and then they release a larva, and the larva is the dispersal propagule. So one of the things that I was thinking after doing my PhD was like, are these traits correlated? So when you are gonochoristic, you automatically are oviparous. So what I did, or what we did actually, was creating a matrix of 3 different genes, and we did this phylogeny to use it as a backbone for mapping characters. So all the taxa that we put in this phylogeny, we knew about the reproduction. That was really important for me, because otherwise you just hypothesize, like thinking about what the reproduction could be. So we had information for the reproduction in all these taxa. And we did this ancestral character reconstruction. So basically what we wanted to do, what would the ancestor of the sponges be doing in terms of reproduction? So if you analyze it using parsimony, you would find that the ancestor of sponges might be hermaphroditic and also viviparous. So these traits are actually correlated. But then these characters are very labile, so they change a lot. And we think, well, if you analyze it using maximum likelihood, you would have a slightly different result. But the major things were the same. So they might be hermaphroditic and also viviparous, and gonochoristic, and then viviparous. So the major traits are correlated, but they can change really quickly depending on the ecological conditions where sponges are growing. So let's talk about what are these traits. How do they look like in sponges? So the first thing I wanted to show you is how oocytes look in sponges. So they don't have gonads. The poor little things are a little bit simple in that way. So they put their eggs everywhere, wherever they can. But sometimes they envelop these oocytes with a follicle. So this is like the first step of having a gonad. It's like you actually separate your gametes from the rest of the tissue. And sometimes you see cells, you can see these arrows there, these black arrows, they have cells that are called Nur cells, helping the oocytes to grow. And how do the oocytes grow? They are constantly making yolk. And yolk is the nutrient, the reserves that the embryo would use during the entire embryonic development, and also the larva, when the larva is like looking for a place to settle, they totally rely on this yolk because they are lecitotrophic larvae in sponges. All sponges have lecitotrophic larvae. So they can make their yolk by autosynthesis, so it's the oocyte itself making the yolk using the Golgi apparatus, or they can have a little bit of help from these Nur cells. So it's basically other cells making the yolk for the oocyte. And this is how it looks in sponges. You can see in the bottom of the picture a cell that is making the yolk for the oocyte that is the rest of the picture. So it transfers the yolk to the oocyte. Or you can see in the yellow arrow, it's the Golgi apparatus, and the orange arrow is actually the yolk platelet being made. But sponges are very weird animals. I learned that from the very beginning of my PhD. So if there's something they can do differently from the rest of the metazoans, they will do it. So they can actually make their yolk using directly the bacteria, the symbiotic bacteria they have in their bodies. So what they do is the oocytes start engulfing this bacteria, they digest it a little bit, they put it together, and here you are, your yolk platelet. That is super quick and super easy. And we thought that was just a peculiarity of this Symvaxinella damicornis, that species that is super frequent in the Mediterranean. We thought maybe it's just this thing that is doing this yolk-using bacteria. Well, no. We very recently found it in another species that grows very commonly in Antarctica. So this is something that sponges can do. This is an advantage they can have because they don't need to wait for this very long process of making the yolk using the Golgi apparatus or the endoplasmic reticulum. They can do it directly with the bacteria. And when they make this yolk, it can be heterogeneous platelet, very weird-looking platelets. So in those platelets, you have the proteins and the lipids all together, so it's very difficult to see which part is the protein and which part is the lipid, but it's a heterogeneous platelet. Or they can do it separately. They can have these protein platelets that you can see here, very dark, or the lipid platelets. And they do it. We don't know how they do it, but we think it might be a similar way to what insects do or what any other organisms do, because they might have the same genes. So that was one thing we wanted to know. Do they have the same genes to make the yolk? Usually, in other animals, what you see is that the genes involved in making the yolk is beta-logenin, cathepsin D, and cathepsin L. So we looked into our transcriptomes and the genomes as well of sponges, and we found that they do have beta-logenin. So that's one advance. But they have two different beta-logenin proteins. One is the one that you see with green, orange, and pink domains. And the other one is very similar to what Siona intestinalis has, which is an ascidian. So there are two different proteins, and we were wondering maybe these two different proteins or enzymes can make different morphologies or different yolk platelets. That is something I cannot answer yet, but maybe in 10 years or so, I would be able to answer. So we looked also for these other genes involved in the yolk formation, and these are cathepsin genes. And sponges have all the repertoires for cathepsins, and actually they have a lot of paralogs in these cathepsin genes, so there's a lot of duplication events in these genes. We don't know what sponges do with these genes, but maybe they have a similar pathway as the rest of the animals. But what I can tell you is that there are some species of sponges that have way more cathepsin genes than the other ones. But we don't know what they do. But maybe this diversity in cathepsins might be related to yolk variability, and this is something that we will be studying. But so far, we don't have a lot of genomic resources, so we need to create or to sequence more to be able to understand this question. But let's talk a little bit about sperm. When I started my PhD, I thought that sperm in sponges, when I was reading like a textbook on sponges or animals, you would read there that sponge sperm is really primitive. And that really hurts me a lot. Because there's nothing in a sponge that is really primitive. It's always something very new. So the primitive sperm cell might be round and probably without an acrosome, and they will be designed, or the shape would be just for floating in the water. And that's true for some species of sponges. You can see that they are round cells, and sometimes they don't have an acrosome, and sometimes they have these proacrosomal vesicles. But that doesn't look like a true acrosome as you would see in any other sperm of any other animal. However, sponges, as I said, can be very weird. And you can see here that sperm can also be elongated in some sponges, and they can have very sophisticated acrosomal organelles. Sometimes some sponges can have this acrosome vesicle and an acrosomal rod and an acrosomal organelle itself. So this acrosomal rod might be involved in injecting the acrosome into the oocyte, so the oocyte can recognize the proteins, the enzymes in this acrosome. And this elongated sperm doesn't occur just in one species. It's very widespread, but it doesn't correlate with phylogeny. So what we found, it's actually that the morphology of a sperm is related to the fertilization mode. Because what happens in these cases where there is elongated sperm is that this sperm is not floating in the water, it's injected into the tissue of the female sponge. So it has to swim into a very viscous medium, so it has to be elongated in order to be able to swim inside. And that's why they have this elongated sperm. And finally, let's talk about the morphology of the larva. Again, if you go to a classical textbook, you would find that sponges only have one type of larva, that is the parenchyma, that it's not a hollow larva, but an entirely ciliated larva. Done. Well, again, that's not true. They have 8 different types of larvae in sponges. They have dyspherulas, synktoblastulas, clavoblastulas. They all look very different to each other, or at least they do for me. And they have this beautiful oplitomela that you have in here, that it's the only larva that can disperse for longer periods of time, because usually sponge larvae can be in the water for maximum 2 weeks. Well, this can be in the water for more than 1 month. And that, for a sponge, is too much. So, but how sponges decide what cell is going to be a gamete? Because I don't know if you know, but sponges have totipotent cells. They are known for having almost the entire body full of totipotent cells, of stem cells that can differentiate into any other type of cell for the sponges. So how do they do it? When they decide that this cell is going to become a gamete. So these cells that will become the gametes are called primordial germ cells. So this is how the sponge body looks like. We have an internal medium called mesohyle, where these stem cells are living, and these stem cells are called archeocytes in sponges. And we have these choanocyte chambers in this left side of the picture, where choanocytes are actively capturing the bacteria that sponges feed on. And these choanocytes are flagellated cells. Well, in sponges, usually these choanocytes are the ones that will become the sperm, and the archeocytes are the ones that actually become the oocytes. But how do they, when they decide, do they decide that they want to make gametes, or how do they decide this? So any choanocyte or archeocytes can actually give rise to gametes, or are there predetermined populations of cells that will become gametes and they're not doing anything else? This is something that actually we don't know in sponges, but even though it's very difficult to track the fate of the cells in sponges, we have other ways. So the first person that started to talk about this problem was Minton in 1900. So he thought that in this larva of Cladrina blanca, the cells in the bottom, in the posterior pole of the larva, that are undifferentiated blastomeres will become something called minute cells, then archeocytes, and then tococytes. But this was just a suggestion. He could never follow the entire process because that means following the same sponge from the larva to the adult. That would mean 2 or 3 years, something like that. You cannot have a sponge for 2 years in a tank, unfortunately. But we have other ways of knowing which cells will become the gametes, and that is usually by sequencing and locating molecular markers. There are 3 molecular markers that you have in here, PL10, VASA, and nanos, that are widespread in all metazoans, and these are usually the markers that people look for when they want to know what are the primordial germ cells. And they are located in an organelle, usually, that it's called the nuage. We have seen nuage in sponges, in spermatogonia of sponges, but also in a couple of other species in the oocytes. So we were thinking maybe they use the same molecular markers to actually determine or segregate their primordial germ cells. These researchers, Linnean and collaborators, they actually did in situ hybridization to look for these markers in the cells. So in the oocytes, these markers are everywhere. And that's weird, because these markers should be in very discrete organelles, so like in very spotty label. And then they disappear, and they become again active or expressed everywhere in the adult. So that didn't really solve the problem. So we took a different avenue, and instead of using a sexually reproductive animal, we used a freshwater sponge that is easy, relatively easy, to culture in the lab, and you can actually hatch it from a gemule. So they use gemules when the conditions in the lakes and in the rivers are not ideal, so they put all their cells, or some of their cells, into a gemule, and they differentiate their cells, so they become stem. All of them are stem cells, and then when the conditions are good, they hatch, and all these stem cells start to be, their fate start to be determined. So we use this system, which is similar to sexual reproduction, but it's not exactly the same. So when we did the in situ hybridization to look for these molecular markers, we actually saw them in subpopulations of archeocytes and choanocytes in discrete organelles. So they were not widespread everywhere in the cytoplasm, but they were around the nucleus where this nuage is usually located, and they were not everywhere. They were just in a subpopulation of cells. This is a major discovery for sponges. That means that all cells in a sponge are not totipotent, only some of them. There are some cells that are already segregated. Even though a primordial germ cell is totipotent, you understand. So some cells are segregated. They will not become any other cell. They will only become gametes. Same thing for nanos. And this, we did it like back in the day, like in 2008, something like that, when I was doing my postdoc in Canada. And it took us forever to submit the paper because we were not actually sure about our findings. Like, we were like, this is so good to be true. It cannot be true. So we repeated experiments, and we sequenced more sponges. And 2 years ago, somebody found the exact same thing in another sponge. So we were like, really? But now it's even better, because this is something that is widespread in sponges. So we know that there are subpopulations of archeocytes and choanocytes that are segregated. So there is germline determination in sponges. We also looked at other markers, not for germline determination, but for sex determination. When you have a gamete, you have to decide you want it to be an oocyte or you want it to be a sperm. So for that, there is not a lot of literature about this, but there are some genes that have been very well characterized in nematodes or in insects. And these are FEM1 genes, and also the MRT1 genes. And you see here, this is a heat map. So when you see a darker color, it means that this gene is expressed more in that condition, and it's always like a non-reproductive sponge and a reproductive sponge. So the only thing that was really interesting for us was what was happening in these DMRT genes, because in Cortithium candelabrum, this is a hermaphroditic sponge. these genes were only expressed when the sponge was making sperm, but not when the sponge is making oocytes. Actually, the sponge is making oocytes all the time. So we thought maybe these DMRT genes, I don't know if you can see it, in planarians, what they do is they promote maleness. So they are sex determinants for male gametes. So we thought maybe this sponge is just by default making oocytes, but there is one time when this DMRT gene is expressed that they think, okay, maybe I will have to start making sperm. We will be in the process of testing this in the future, but this is promising as well. So let's take a break, because now I'll be talking a little bit about what are the ecological implications of having one type of reproduction or the other one. So coming back to this yolk, you remember now that sponges can have heterogeneous yolk with both protein and lipids, or homogeneous yolk with these platelets separated. But it's totally different to have more lipid yolk or more protein yolk for your embryo, because lipids act as a rapid fuel for active cell division and differentiation. So if you have more lipids, that would mean that you have an ecological advantage once you settle, because it will allow you to grow faster. So we wanted to test if that is the case for some species of sponges. And we had this beautiful sponge in Antarctica that is called Micalia cerata, and is among the fastest growing sponges in Antarctica, and I can say everywhere else as well. It's not extremely common, but when it's common, the whole wall and the whole boulders are absolutely covered by this sponge, and it can be my size, which is not very big, but for a sponge it is. So we wanted to know if larval yolk type may play a role in facilitating early settlement. But of course, we couldn't do it just by looking at the sponge. We wanted to do it in a comparative way. So we took another sponge, Micalia laevis, which is growing on corals. It's a very common tropical sponge. And we wanted to look at the embryonic development in both these sponges. So what we found was really interesting. So in Micalia cerata, all the yolk is lipidic. Like, there is not a single protein platelet in Micalia cerata. So I don't know how they do it. I think what they are doing is they are ingesting, or they're digesting a lot of diatoms, which are very full of lipids, and this is how they get all this material to make their yolk. And we calculated the energetic signature in 8.23. I don't even know to say this number in English, but you see it, right? 8.23 something kilocalories. And then we compared this to the yolk in Micalia laevis, in both the embryo and the larvae. And we estimated that based on their yolk being separated, they have two different platelets, lipid platelets, but also protein platelets. So we calculated the energetic signature in these larvae to be 1.17 something kilocalories. That is a major difference. So it's like 7 times less than in the other species. So we think that when the settlement times come, this Micalia cerata has totally an ecological advantage, because it can rely on all these lipids to start growing really fast. And that gives it an ecological advantage that this poor other little thing doesn't have. And this is the final part of the talk. I'll be telling you about the influence of sexual reproduction on population connectivity. Again, it is in the Mediterranean, which I'll be telling you about sponges from the Mediterranean, because I'm from Spain, so that was close, and I could go to the sea and collect them very easily. So in the Mediterranean, sessile invertebrates with short larval duration phases are less influenced by oceanographic discontinuities, but are highly affected by isolation by distance. That means that two populations that are separated by 10 or 20 kilometers will be absolutely different. That doesn't happen in fishes, or doesn't happen in dolphins, or things like can disperse really, really far away. But sponges cannot do it. But all the bulk of the knowledge of these Mediterranean sessile invertebrates is basically cnidarians again. They're everywhere. So we wanted to test whether this thing is applying as well. This statement applies as well to sponges. Because if you have very differentiated populations, it means that they are more vulnerable, because they will not be able to recover from a die-off or from a disease, from migration from other areas, because the larvae could not get very far. So we looked at two different sponges, two species of sponges. One is Yersinia fasciculata, who is not very beautiful, sorry, and the other one is Petrosia fittiformis, which is actually a very beautiful sponge. And these are very common and abundant sponges in the Mediterranean. If you go dive in the Mediterranean, you for sure will see them. But they are suffering mass mortalities, both of them, caused by global warming, caused by bacteria that are more active. They are pathogens, super active when the water is very warm. So we were really interested in knowing what happens with these sponges, with the connectivity of these sponges, if they have different reproductive traits. They do have very different reproductive traits. Yersinia fasciculata has this larva that is a tufted parenchyma that you see in the left side of the slide. They are what we call in the sponge world as larvae with high swimming abilities. That means super slow larvae, but still for a sponge, they have high swimming abilities. And what is really, really important is that they can be in the water column for about 2-3 weeks. That is a long time for sponges. And then we have Petrosia fittiformis with this crawling larva that is really slow, and it doesn't go anywhere. Like it's basically when you see this larva, it just goes down to the bottom of the sea, and then it crawls for about 2-3 days, and that's all. If it finds like a spot, like a good spot to settle, it does. If it doesn't find a good spot, it will die. So what we did is we collected these sponges all across their distribution range, or as far as we could go, and then we sequenced them. We sequenced these different populations, and we genotyped them using microsatellites, using molecular markers. And then we assessed different parameters. So one of the first things we wanted to assess is the clonality. Again, if you hear about sponges, you will hear that sponges are clonal organisms. That means that they can regenerate or they can reproduce by asexual reproduction really easily. Well, that's not true for a lot of sponges. You see here, they have very low clonality levels. In 200 individuals of Yersinia fasciculata, we didn't find a single clone. And in Petrosia fittiformis, we had 300, and we found 4 clones. That is almost nothing. And this is because they are massive sponges. So encrusting sponges, when a sea urchin goes over them and splits the sponge in 2, then you can have 2 sponges really easily. But they cannot do the same thing with these massive sponges. Petrosia can be this big. And it doesn't fragment very easily because they are really hard. So they cannot regrow once a fragment is gone. So they mainly rely on sexual reproduction, these 2 sponges. And then we found that they have 3 clusters of populations, and they are, I'll show you, they are located in very similar areas. So there are major genetic discontinuities that we think are correlated to oceanographic discontinuities, so fronts, actually, oceanographic fronts, that are creating 3 different areas in the Mediterranean. And we wanted to see whether this larval type would affect their differentiation. So in this sense, you compare two populations, like every time you compare 2 populations to each other. And then you can see if the FST, which is the statistic, is significant, then you have 2 different populations that they don't mix really. So there's not migration or there is not gene flow. So in Yersinia fasciculata, we saw that all populations are significantly different, almost all, except for 4. So almost all populations are significantly different. But the maximum value, this FST value, was 0.2. I'll show you. In Petrosia fittiformis, the maximum value of differentiation was 0.4. It doesn't matter what it means. It's double the value. And I grew tired of putting asterisks in this Excel file, but all of them are significantly different, all of them. So that means there's something that is making these Petrosia fittiformis populations differentiate massively from the rest of them. And if you put that in context, these 2 pink dots in there, and there are other sponges from the Mediterranean and from other areas, and you see there are major differences. And we think that these differences are caused by these 2 larvae that they have. So crawling larva of Petrosia fittiformis, it doesn't go anywhere. So you have 2 massively different populations. But this other sponge, this Dictyoceratid, has high swimming ability larvae, and they can more easily connect their populations. And just to finish this talk, I'm going to tell you what are we going to do now. In my lab, we thought, like shallow water species are so easy to collect. Let's take a challenge. Let's go and collect them in the deep sea and see if they do a very similar thing or if they do different things. So we have this project that is called Sponges, very creative acronym, I guess. And it's a European-funded project. And we are 20 institutions in Europe, and this is a very multidisciplinary project, but we take care of the sexual reproduction of these deep sea sponges and also how it affects the population connectivity. So my students and my postdocs, I'm not doing anything, they do all the work. They are doing description of gametes of these deep sea sponges because we know nothing at all. We are doing differential gene expression during reproduction to see what are the genes they use for making the gametes. They're doing jock analysis as proxies for larval duration phase because we don't even know if they have a larva or not. We are doing population genomics, and we are doing something very interesting that is symbiont vertical transmission. So sponges can put their symbionts in the oocyte, and that is how they transfer these symbionts to their progeny. So we want to know how they do it in deep sea sponges. And that was all. Thank you very much.